اتفضل
صباح
دكتور.
الخير، صباح الخير المستمعين. Good morning dear college. أولا أنا اسمي علي الخان، باحث بالقمح في تركيا. My name is Ali Ilkhan. I'm reader, Witty reader from Turkey. Dear college, I'm very glad to be with you today. First of all, I would like to express my sincere gratitude to the Arenina team for entrusting me with the coordination of the Plant Genetic Resource Network. I would like to emphasize that I will do my utmost to strengthen this network and make
ايوه
it more active and
اي
effective
والله ماشي الحال
platform
فتحت الكمبيوتر.
for regional collaboration. It is now my honor to briefly introduce our distinguished speaker, Dr. Hüsnü Aktaş. He is a highly respected scientist with extensive experience in wheat breeding and genetic resource, particularly in the Southeastern Anatolia from Turkey. Throughout his career, he has contributed significantly to numerous national and international projects and has played an important role in the development and registration many wheat varieties adapted to region. Yes, today he will present on the potential of the Southeastern Anatolia region of Turkey in terms of wheat genetic and research and their sustainable conservation.
دكتور علي، بس عفوا ممكن ترفع صوتك شوي؟
نعم.
بس ممكن ترفع صوتك شوي؟
آه.
الله يكرمك.
I would like to thank again to Dr. Hüsnü. I'm advanced for his valuable presentation and now invite him to take the floor. Dr. Hüsnü, yes, please.
Okay, I can start, Doctor.
Yes.
Okay. Dear scientists, dear colleagues and participants, I am very pleased and honored to be here with you at this webinar on such important topic as biodiversity. I believe that this meeting will make significant contribution our knowledge. I would like to thank everyone who contribute to organization, and I wish a productive meeting. I respectfully greet you all. The first, I want to introduce myself shortly. I am Hüsnü Aktaş. Now I am working on Mardin Artuklu University, Department of Field Crop. Before I was working on the Research Institute of Diyarbakır. For 18 years, I was starting as a technician, and then now we are here. I have been in CIMMYT, ICARDA, and I have several colleagues. We now talk in some time. I think we know each other, some of participants. I don't know, because my screen now, I cannot see you. Just I'm talking. I will start. Our topic is potential of Southeast Anatolian region of Turkey in terms of the genetic resource and their conservation. In screen we can see how is genetic diversity. We can understand just from this screen. You can see several different spikes that collected on our region, Southeast Anatolia. Also, some picture you can see the Göbekli Tepe. I think you hear and you know. Also is located in our area. Our presentation outline you can see. importance of the wheat genetic resources. Karacadağ, I will mention, this area is very unique for genetic resources for wheat. Purpose of using wheat genetic resources, examples of use of wheat genetic resources, land races with varieties of the Southeast Anatolia, some conducted study and some recommendation. As you know, our region is located southeast of Turkey. Also, Turkey is located between Asia and Europe continent. Also, for genetic resources, very important area. Our region is between Euphrates and Tigris rivers. Also, you can see the Göbekli Tepe here. Those rivers has 28 of water source of Turkey. These two rivers also important for all the area, also for Iraq, Iran, for Turkey. Also, these rivers very important for civilization of the human and start to agriculture activities here. Everyone, I think, here know the Fertile Crescent, Mesopotamia. This region is part of the Mesopotamia. Also very important origin and on domestication of the wheat. Wheat was cultivated in this area, or Karacadağ mountain, first time in history. Why are plant genetics so important? We can say these plant genetic resources have range variation in gene alleles that confer resistance to exist stress condition and potential adverse condition. Genetic resources also shaped by the long-term adaptation to stresses and serve as safeguard against today's challenges are recognized in many international agreements. The three main principles of the United Nations Convention on Biological Diversity, most comprehensive of this agreement, conservation of resources and their sustainable use, and the fair sharing of benefits arising from their use. We can classify genetic resources as a wide space, for example, for Dicocoides, Boeticum, Taushi, translational form, Dicoccum, Monococcum, land races, and old varieties, for example, Sorgül, Havrani. I think Havrani also in Syria, in Iraq, known also in Turkey, there are these varieties still. And the underbreeding lines were improvement lines or varieties. For example, Qaus, Pasteur, Sham 1, one of them. From past to present, you can see the genetic diversity decrease. There are some reasons because of the uniform food consumption. We can say everybody in different country now consumed işte hamburger or pizza. So the uniform food consumption decrease the genetic resources, natural resources also, extensive use of natural resources, agricultural activities, especially we use the herbicide or pesticide, and we kill the all genetic resources, especially for wheat. Urbanization, you can see the population increase and new building in every city. Also, plant breeding activity effect on the losses of the genetic resources. When we make the breeding, we development yielding varieties, and then the farmers don't prefer the use land races. So we lose our genetic resources. Genetic resources conservation method, there are three methods. In situ conservation, this conservation in natural habitat. Which one is more efficient? We can say that in situ conservation allows population to continue evolving under natural selection. So this is very useful for us. The another one, ex situ conservation. Genetic resources are conserved outside their natural environments in gene banks or gene research garden. The another methods, biotechnological methods. Fertile Crescent, as you know, this area where many carols, legumes, and other crops used in human nutrition were first domesticated. Also, I want to mention about Karacadağ. Karacadağ is very unique area for wheat resources. Many researchers who have studied wheat evolution in Southeastern Anatolia region of Turkey have reported that Karacadağ is seat where the wheat was first domesticated. Karacadağ is not only volcanic mountain, but also basalt covered region located in triangle of the Diyarbakır, Şanlıurfa, and Mardin province in Southeast Anatolia, covering an area 120 kilometers in diameter and 8,000 kilometers square. It's very big area. Why is genetic resources diversity is very high here? In this area, genetic diversity is mainly due to variation of topography, climate, other environmental condition over short distance. I work on this area when we start from, for example, 500 meter altitude. After 10 kilometers, when we use the one of the GPS, we see that we are on the 1,500 meter altitude. So for short distance, topography and altitude is changeable. Because of that, genetic resource diversity is here very high. When we see the origin of the durum and bread wheat and Karacadağ, we know that wheat is have three genome, A, B, and D. B and A crossed each other naturally, and wild emmer Triticum dicocoides occurred, and then transitional form dicoccum, and then durum wheat cultivated. After that, how the bread wheat is occur? Triticum dicoccum or dicocoides crosses between Aeligos taushi and then spelta and bread wheat is occur. So why we can say in Karacadağ three genome that have three spice grown here naturally: Speltoides, Urartu, Dicocoides. So we can say that not from the some agricultural event. Because of these three spices grow here, we can say that the origin of durum and bread wheat is Karacadağ. We can on the screen Triticum urartu and Speltoides, and then the cocoides occur, and then a cross between taushi and bread wheat exist. The cocoides convert to dicocum, and dicocum convert to durum. Yes, the another one, Triticum beaticum or urartu, convert to monococcum. These two, dicocum and monococcum, translational type. This wild or translational spice have rapid grind feeling. Iligos peltoides show strong competitive against to weeds, also resistance to herbicide. For example, when we use the herbicide on the speltoides, we cannot kill the speltoides. Maybe these spice have a natural resistance to herbicide. There is some challenge or threats to genetic resources in Karacadağ. For example, conservation of rangelands into the crop plant through the polling and overgrazing. Straff burning, intensive use of the fertilizer or agrochemicals, and spread of the yield cultivars. Urbanization, industrialization, and construction of the roads and dams. The use of basalt stones that naturally protect wild plants for gravel production. Also, removal of the astragalus, which provides natural protection for wild plants for use as animal feed. You see this astragalus plant and wild type of the weed live together. When we removed this astragalus, the habitat of the wild weed not nature anymore. This photo also from the Karacadağ. This one is Triticum dicocoides, one of Boeticum, and all the area covered by basalt stone, and this product these wild weeds. Also here there are animal activities, feeding and over grazing still is continue. This photo from maybe 10 years ago, I was working on the Karacadağ. We collected the wild spice of the wheat. About 1 month we collected, but just one part of the we can visit and collected wild variety, wild spice from Karacadağ. Also in Karacadağ, the stone is collected to construct building or for road. So this natural area now is broken. Also, this stone not just for plant, also for wild animal is important. So this challenge still is occur. I think it is not good for our diversity, these activities. Also, we collected from all the Turkey the wild spice of the wheat. Most of them, you can see from map, located Southeast Anatolia. So Dicocoides, Boeticum, Hulutwitz, Timopevi, Urartu, Taushi, all grow naturally in Southeast Anatolia. Purpose of utilization of the genetic resources. You know, the genetic resources are used in breeding programs to transfer desirable traits into the modern cultivars through hybridization or biotechnological approach. Also, we can directly use these resources. Genetic diversity is essential for development of the genotypes with these traits. While with local alternative races with genotypes exhibit genetic diversity in terms of the RHT gene. For plant height, we need different RHT genes, vanillization gene, photoperiodism gene, also for quality, high molecular and low molecular glutenin alleles, also as well as for yellow rust and leaf rust genes. These resources have again a big diversity. For example, coleptil length. Land races have a long coleptil length, but the breeding lines coleptil length is shorter and not suitable for deep sowing modern wheat varieties. Also, vanillization gene and photoperiodism sensitivity genes we need for adaptation of that we developed varieties in the wild, primitive, and local old land races with we have a very big diversity for these two traits. Also, grain filling rate is very high and very fast in wild and primitive wheat. So to escape the some stress condition, we need fast grain filling rate. Also resistance to fungal disease, novel high molecular and low molecular glutenin alleles. We need these land races white and primitive with genetic resources. Reducing plant height also is important. You know the RHT 8 and 9 you can see the on local genotypes or the primitive or wild types. These two genes decrease the plant height, but don't affect on the colectil length. So these two traits we can see on the land races, wild and primitive wheat. One of my work, land races, colectil length, we see the very long compared to breeding lines. 1, 2, 3, 4. This the left side is breeding lines, the right side is land races. Just one example for colectil length. Examples of direct use of the genetic resources and potential of land races. You know the development of the semi-draft varieties in Italy first using Acomigo cultivars, which carries RHT8 and PPD D1 intensive genes brought to Japan after Korea Japan war. The removed Nobel Prize winning with breeder Norman Borlaug. Norman Borlaug transferring drafting genes into the wheat. Genetic resources called Norin 10, native to Japan, and carrying the plant height reducing genes RHT1 and 2 was utilized. The event known as the Green Revolution was likewise achieved through the use of genetic resources. You can see the picture. Land races have a high plant height, but RHT1 and RHT2 transferring the land races and plant height decrease. Before the Norman Borlaug, Italian scientist Nazareno Strampelli tried to reduce the plant height. But Norman Borlaug have a more efficient work and then win the Nobel Prize. Another example, synthetic wheat varieties. I think maybe you hear normally in the nature we have bred wheat land races. But by human made, we can Triticum durum and Taushi cross each other, and after doubling chromosome, we can handle synthetic hexaploid wheat. Why you use the Taushi? Because the genome diversity is very poor for most of traits, so we try to gene pool of the D genome more range or want to decrease genetic diversity for gene D genome. Also,
Dr. Hüsnü, sorry,
yes.
Last 5 minutes.
Okay. The another examples of the direct use of genetic resources, potential of local varieties, one of them Turkey wheat varieties. You know, the Ottoman Empire, some settlers migrating from the Ukraine and from Ukraine to the Nebraska, and these settlers give the Turkey wheat Kansas and Nebraska before from the winter hard condition. Just in America, spring sowing they can do, and yield is very decreased. After the wheat, Turkey wheat arrived there, they could sowing in the winter time, and the yield is doubled. Also, there are still stated Turkey wheat varieties in America. Another example, Honorec, Saragola, Manitapo, is land races of the Turkey, but in Italy or Switzerland, they contribute to the breeding program. Another example is Sardari from Iran. Sardari is, how I say, both under cold and drought condition, the long coleoptile and root system. This variety may be the reason for its tolerance, drought and cold stresses. Most cultivated variety in Iran still is land races. Colored is Sardari. Another example, Kunduru, Aşure in Turkey, Sorgül, Havrani, Bağcak, one of them. You can see some scientists, Vavilov, Dr. Harlan Zikovsky, but one of the scientists, Mirza Gökül, I want to, this scientist is very important for wheat land races for Turkey. According to the report of the Gökül, we lost most of our land races. Before I worked on treaty project for FAO. From all Turkey, we collected land races, but most of them we collected from the Southeast Anatolia, and genetic diversity very high compared to the another part of the Turkey. Some land races, you can see the compactum. Now, for biscuit industry, prefer this spice, but very unique for biscuit, I can say. The another one minute, doctor. With general characteristics of the farmers practicing local wheat cultivation and protect this area, the farmers with limited financial resource, lacking agricultural technology, no or limited herbicide, insecticide, or fungicide. Also in areas where livestock raised for straw production, generally by older family for household consumption. Are there any risk for trees for future of the land races? The younger don't interest this issue. Young people living in cities and migration from the rural areas. The split of the high-yielding modern variety also is a problem. Extensive use of the pesticide. Also, how we can production of the local wheat varieties be made sustainable, providing government support for production of land races, attracting the interest of the industrialists. Conservation activities should be carried out under farms condition, efforts to increase the market value of the local varieties, incentive for expertise engaged in local seed production, establishment of pre-breeding programs in agriculture institutes and universities. Some studies I conducted diploid white originated from Turkey, molecular characterization, also using functional markers and their validation to determine variation during wheatland race in Southeast Anatolia. Also using functional markers and their validation, also we work with Dr. Alex Murganov. We have also Hulud with book. You can see it on the internet. Some photos are my activities before 10 years ago. You can see some meeting for farmers, with farmers. The last, I can say that Karacadağ, land races and wild genetic resources are our future. Thank you for listening.
Thank you, Dr. Hüsnü. Thank you for interesting and very important presentation. Have any question, dear friends? Yes. Can you asking English for and also Arabic? We can translate to Turkish, to Arabic, to English. Yes, Michael Baum, Dr. Michael Baum.
Yeah, good morning. Thank you very much for the interesting presentation. My question would be, have you done any systematic evaluation of the germplasm collected? You know, the gene banks, they have the possibility, I mean, they do what they call a gap analysis because, you know, how many accessions you need to collect, for instance, a certain species. You know, so they have like an optimum number. Let's say, you know, if you have 5,000 accessions for a certain species, that might be sufficient to have whatever 95% of the genetic diversity collected. So, are there any some of these systematic studies to understand if how much of the genetic diversity of this important region has been already collected and preserved in gene banks, and how much still needs to be done?
Yes, Thank you. Dr. Baum, as I mentioned about Karacada, you know the very big area. We try to collect it, genetic resources of the wheat from Karacada, but for example, we studied for content of the mineral content for example zinc, iron. We used 200 genotypes of the, for example, for Boeticum, or 3,000 100 for the Cocoides, but we cannot collect it all the, 1,000, 3,000, but I know that gene banks from Turkey every year try to collect these wild varieties, but I don't have any information about gene bank. We know that the gene bank have several collection there, but our funding support just enough for the 200, 300 analyze it or something like that.
Yeah, but it's really important that that these collections are done systematically, because
Yes.
maybe you have already sufficient. Like for instance, if you look at the Ikada gene bank in Lebanon, between Lebanon and Morocco, have 40,000 accessions of wheat. Yeah, of course, it's that they are collections from the whole region, and so maybe there needs to be some discussion between let's say Ikara gene bank there in Lebanon and some gene banks in Turkey to see if the important diversity has been already collected, and that also can direct future collection mission, because maybe I'm not saying I have no idea. It's not exactly my field, but maybe there is already sufficient diversity collected, or there need to be more collected, but then it needs to be directed. So to know what's missing in terms of species, in terms of traits, and so on. So that's why there needs to be some kind of systematic analysis what's already available.
Yes, you are right. Sometimes there are some challenge between, gene banks and the another gene banks. Sometimes we try to allow the collected, wild spice, but allowing the processes, bureaucracy also is very, difficult. We try to our best, but I don't know. You are right.
Thank you, Dr. Michael. Thank you, Dr. Hüsnü. Yes, Dr. Ayman. Dr. Ayman.
Yes. Assalamu alaikum.
Alaikum assalam.
Good morning for everybody.
Good morning.
First, let me thank very much the presenters for such a great topic. In fact, as with genetic resources are nothing but the biological foundation for the food security. I have two questions, if you allow me. The first question that we know that the wild relatives of wheat are vital for adaptation to extreme harsh environmental conditions, but unfortunately they face severe threats. As we know that approximately 16 to 35% of them are threatened with extinction, and an even larger proportion is experiencing genetic drift, or what we call a genetic erosion. So conservation, whether the ex situ or in situ conservation are unfortunately insufficient. So my question is, given that 95% of wheat wild relatives are not yet collected, what are the recommendations for prioritizing urgent collection missions before habitats are lost? The second question, sorry, all we know that land races are often less productive than modern varieties. I mean, the limited number of high-yielding varieties which are focusing by the genetic programs. We develop high-yielding varieties rather than cultivating the land races. But unfortunately, the production capacity of the land races is much lower than the modern varieties. So my question is, who should bear the cost of maintaining wheat genetic diversity? Do we need new economic models like biodiversity credit or subsidies for on-farm conservation to prevent the continued erosion of the land races? Thank you very much.
Thank you, Dr. Ayman.
Dr. Ali, I try to understand two questions, but for wild relatives conservation, yes, the losses of the, I mean, collection or genetic diversity, I don't know how much, how percent. I mean, for my region, but I can say that very big fabric and dam or several things founded in this area. So we can say that today, still in this area, we have genetic diversity here, but maybe 10 years, after 10 years, maybe 20 years, we can lose all of them. So because I don't know, of course, the governments should protect, or Minister of the Agriculture should some, I don't know, some politic or something. But I know that, or I guess we can maybe 20 years after, just we will have on gene bank this, I mean, wild varieties in Karacadağ, for example. Maybe we cannot see any other. We cannot see in that area these resources. The another question, land races. Yes, land races yield potential is low. In presentation, I said that some supporting we need for government or for Minister of Agriculture. We try to make some crosses between land races and breeding lines, also land races to land races. Why? Because when we crosses between breeding and land races, still plant height is high, but some genes, I mean, when we cross between land races, not dominant genes. The another one, how we say, we can between land races, plant height can be the decrease because of the some genes is we can how I say collected in the land races, between land races.
Okay. Okay.
Thank you, Dr. Ayman.
Thank you.
Dr. Firas. Yes, please. Dr. Firas.
Okay, good morning, everyone. Hello, are you hearing me, Doc?
Yes, yes.
Yes, yes.
Oh, okay, thank you for your good and excellent presentation. My question is, you are focusing on biodiversity or breeding programs and how biodiversity of wheat. My question is so far from this. I don't know in Turkey if you did any breeding program against different kind of insect, whether for genotype, accession, land races, or cultivars of wheat, because this is so important point. And the second point, I don't know if these genotype, accessions, or cultivars or variety of wheat will be tolerate the accelerate or rapid global climate change in order to distribute it from Turkey to another countries in the Near East or North Africa. I'm the coordinator of Plant Protection Network at Arena, and thank you.
Thank you, Dr.
Yes,
Firas.
thank you. Let's to I will. Yes, we made some work for characterization of the land races that we collected from all of Turkey. We using functional markers, and the validation to determine variation in bred wheat and durum wheat land races of Southeast Anatolia. We use some markers, adaptability genes, for PPD vernalization genes, ground size, weight, and yield related genes markers we use, drought tolerance, and there are another for traits, but still we try to how I say publish in some journal, but we make this work that we collected from land races from all of Turkey, very big. How I say the collection. Also, we give this collection to Gene Bank of the Turkey.
Thank you, Dr. Firas. Thank you, Dr. Hüsnü. Last two questions. Dr. Rasha, please.
Assalamu alaikum. Good morning, everyone.
Good morning.
Thank you for the nice presentation. My name is Rasha Tarawne. I'm the Dean of Faculty of Agricultural Technology at Al Ahliya Amman University in Jordan. And we are in our faculty interested in the genetic resources and especially what I'm doing in my what I did in my PhD and doing now in my research, working on barley and wheat resources from different origins, different countries. I have just one questions about the need for genotyping. I think with this rapid loss of these traditional land races and wild relatives in Turkey and also in Jordan and in many countries, I think we have an urgent need to genotype this collection or the genotypes. So I don't know if in Turkey Gene Bank or in your institute you start to genotype these some of these accessions.
Yes.
And also I want to tell you something that I got the opportunity to do my PhD at the German ex situ Gene Bank in Germany at IBK Institute, and they have a large collection of wheat from different countries, and they have a good collection from Turkey, and also these collections are genotyped, and maybe you can connect this compare the collection you have with the collection they have also, and you can use and translate the phenotypic data you have with the genotypic data they have also, to do some collaboration in this area. Thank you.
Thank you, Dr. Rasha. Yes, you are right. We have connection for gene bank of Turkey. They say we genotyped this collected, collection. Also, you know that the some how I say collaboration between, for example, Lebanon and Turkey gene banks. I'm working on the the university, but before I was working in the research institute. That time as very easy when I was in the research institute to connect gene banks, but from university to research institute now is little not easy like I was in institute, but we can collaboration with you. Also, we have some collection barley, for example, how I say Gordeum bulbosum. I remember. Or land races still I have. Also, I conducted one barley land races for master thesis with my student.
Yes, thank you, Dr. Hüsnü. Last two questions, I'm saying Dr. Marwa and Dr. Amri, and and offer the presentation. Firstly, Dr. Marwa, please.
Good morning, everyone. Thank you for the nice presentation. I'm Marwa Hassin, a cereal breeder in Tunisia from the regional center of field crops of Beja. I would thank you again for this presentation and very interesting meeting. I have just one question. I'm actually working also on line races in Tunisia. We have a huge collection also in Tunisia, and in this last period, we make a sequencing for just for 2 land races. It's very important for us and all for all the country in Mediterranean region due to the climate change. My question is, did you use these land races in breeding program for increasing quality traits and plant disease, especially actually, the climate change have the impact in the yield, but also on nutritional value of our improved variety. As you know, the land races are very known for their quality traits. That's why I'm asking about if you integrate these land races in breeding program for quality and plant disease. And thank you so much.
Thank you, Dr. Marwa. We try to make crosses. We make crosses between land races and breeding lines, but the problem was still, for example, F3, stage F5 and F6, after the homozygote, F6, we see the still plant height is high. So for big challenge is plant height still. We mentioned some dominant genes still. But still have. Yes. So also my recommendation in presentation, I said we all the, institute or we start to pre-breeding program. We don't have pre-breeding program. So, a pre-breeding program and then breeding program. We should follow this. So we don't success. We are not success. We tried to, but I don't know. Yes.
Hopefully, good luck. Thank you.
Thank you, Dr. Marwa, Dr. Hüsnü. Yes, Dr. Ahmed Amri, تفضل, please. Dr. Ahmed Amri. I think he is not here. Again, Dr. Rasha, have you any question? Dr. Rasha?
You forgot your hand raised.
Me?
She has no question, I think.
Yes, Dr. Ayman. Dr. Ahmed Amri. Yes, please. How are you? Open voice, please. Dr. Ahmed.
دكتور أحمد بس عفوا أنت ميوتد، ممكن تعمل أنا ميوت؟
مش مفتوح voice. دكتور أحمد. دكتور أحمد. We don't hear.
دكتور أحمد ممكن تعمل أنا ميوت للصوت؟
يا دكتور أحمد افتح المايك إذا سمحت، افتح المايك.
I think he's have problem with the voice.
With connection, I think maybe voice maybe is overload is not working.
I think
For Ahmed Amri.
a problem. Yes. Dr. Ahmed Amri, connect it again, please.
I know Dr. Ahmed Amri from Ikarda. I think we see each other in Turkey.
Yes.
Also in Ikarda.
Michael Baum offered to Dr. Ahmed Amri. Can you write to us, Dr. Ahmed? Sorry.
Because we cannot hear you, Ahmed. You are muted.
But they
دكتور رايا.
دكتور أحمد بس عفوا إذا ممكن تفتح المايك، أنا بحاول أعطيك إنه ask to unmute بس أنت ما عم تعمل unmute.
شو نعمل؟
إذا ممكن دكتور أحمد ممكن تكتب على الشات السؤال أو المداخلة.
Can you send a WhatsApp message, a voice message on WhatsApp?
Or in chat.
Maybe. So Raya,
Okay.
Dr. Ahmed can he write to mail,
Okay.
email. Then Dr. Hüsnü's answered his question. Okay. Thank you.
Okay, Dr. Ahmed, you can send your question by email, and I can forward it to Dr. Hüsnü.
Yes, thank you to
Yeah.
all participants. See you at the next meeting, inshallah. I hope you are very happy. The next month الشهر الجاي إن شاء الله نتلاقى ثاني موضوع دكتور رضا شبلي. Thank you very much to all the participants. Thank you very much.
Thank you for your thank you.
Bye. See you again.
Bye.
Thank you everyone. Bye.
Thank you
Very good.
very
مع
much.
السلامة.
شكرا. Thank you very much.